It's the time to have the next invited speech given by Dr. Reza Rajimehr. Dr. Rajimehr, hello. Hi, Reza.
Hi, so should I start?
Just a moment. It's nice to see you again, Reza.
Yeah, nice to see you.
Yeah, okay. I will brief you a little bit and then we can start your presentation. Okay, I know Reza from 20 years ago. It's nice to see him again. Reza, Dr. Rajimehr has obtained his medical doctor from Iran University of Medical Science, and then he has a PhD. He did his PhD in University of Cambridge, and during his medical school, he was a researcher in IPM when he started his research in cognitive neuroscience. And then he continued his research in Massachusetts General Hospital of Harvard Medical School and MIT later as a postdoc and research scientist. His study actually mainly focused on the functional organization of the visual cortex, and mainly Reza is using Fmri techniques in his study. He's an expert in Fmri data collection in, I think, human and monkey, and also in the analysis of those data. Okay, Dr. Rajimehr, we are looking forward to have your presentation, which is about semantic phase processing in human brain.
Okay, thank you, and also thanks for inviting me for this symposium. So today I'm going to talk about a study that I did with Neda, who was PhD student in IPM, and she has graduated now. And this is the work that we did together in IPM. Okay, so as you know, face recognition is a very important task in everyday life. It plays an important role in social communications. Face processing in the brain is carried out by a network of cortical areas. Classically, these areas are classified into two groups. One group is called a core network of face processing, which includes regions in ventral occipitotemporal cortex. These regions include occipital face area, fusiform face area, and posterior STS face area. STS means superior temporal sulcus. So these areas basically do visual analysis on faces. Basically, they extract information about facial features, facial configuration, the viewpoints of faces, facial expressions, and also they do processing of viewpoints and also identities of faces. In addition to these core areas, we have another set of areas which are called extended network of face processing. These regions are usually outside of ventral occipitotemporal cortex. They are located in parietal cortex, particularly medial parietal cortex. Some of them are in frontal cortex, and also we have some subcortical structures like amygdala. And these regions are believed to be involved in processing of more high-level aspects of faces, such as social information of faces, semantic information of faces. And actually, the talk that I'm going to give is more focused on this extended part of face processing network. So one form of face processing is familiar face processing. So when you see a familiar face, what happens in the brain is that a cascade of perceptual analysis of faces is followed by a process of retrieving semantic information or semantic knowledge about faces. So it is semantic because in order to recognize a face as a familiar face, you need to basically check your memory. You have to compare the face with your memory representations so that you understand that this face is a familiar face. So basically, you need to rely on some semantic representations in your memory in order to understand a familiar face. And at the same time, once you recognize a familiar face, that usually brings up some contextual associations. So for example, when you see the face of Barack Obama, so you know that he's a politician, his wife is Michelle Obama, he made a nuclear agreement with Iran and many other information that are associated with this face. And the activation of those contextual representations also basically they're also considered as part of semantic processing. So I will come back to this slide, but let me say a little bit about the neural substrates of familiar or famous face processing. So familiar faces are processed in a distributed network of cortical areas and also subcortical areas. These regions include inferior frontal gyrus, anterior temporal lobe, medial temporal lobe, amygdala, and posterior cingulate cortex. If we look at a meta-analysis of familiar face processing, we see this set of regions. I just named them in the previous slide. So for example, we have anterior temporal lobe, we have medial temporal lobe, regions in inferior frontal gyrus, and also one of the main spots of familiar face processing is posterior cingulate cortex. So what is the goal of our study? So let me come back to this slide. So when you are seeing a familiar face, sometimes you extract multiple information about that face, and these information are relatively hierarchical. It means that you basically recognize a face as a general category of faces compared to other categories. So this is a very general level of face recognition. And then at the next stage, you recognize it as a familiar face. And then you also recognize the occupation of this face. So he's a politician. So you assign this face to a specific subcategory of familiar faces. And finally, you identify this particular face, who is Barack Obama. So basically, in this process, you have multiple stages of abstraction of semantic information. So the question is how these multiple levels of information abstraction are represented in the brain, so where they are represented, and if there are representations for different hierarchical levels of familiar face processing, whether those representations have a specific topographic organization or not. So these are the questions that we are going to address in this study. So we did an Fmri study to address these questions, and in this study we used a set of faces. Basically we used 40 face images from 5 categories. 4 of them were familiar faces, like 4 of them were familiar face groups or face categories, including faces from cinema group, music group, politics group, and sport group. So all of these faces in the upper row are actors. The next row, they are singers, and then we have politicians and football players. So we had 4 categories of familiar faces, and also we had 1 category of unfamiliar faces. We made sure that our subjects can recognize these faces. So we had a questionnaire. We assessed their behavioral performance. So we asked the subjects to say whether this face belongs to cinema, music, politics, sport, or unfamiliar group. And based on these confusion metrics, we made sure that our subjects can categorize all of these faces into 5 categories. And then we are going to use these stimuli in an fMRI experiment. And because we are going to do a multivariate pattern analysis to see which parts of the brain contains information about these faces, it's quite important to control for low-level visual features. Otherwise, if you don't do that, you may find a region of the cortex or brain which can classify these faces, but that classification might be just because of low-level factors. So it's quite important to control for low-level features. So we did a very careful matching. We matched our stimuli based on brightness, contrast, size, luminance histogram, and power spectrum. We used the Shine toolbox for this equalization process. And once we matched our stimuli, when we gave them to a V1 model and asked the model to classify the face images into 5 groups, the performance of the V1 model was basically at the chance level. So these face images were not discriminable based on low-level factors. And the reason that you see some patterns in the background is because of this equalization process, mainly because of matching the power spectrum. And then we had an event-related fMRI design. So in this design, we presented individual faces followed by 1 second blank period. And then we presented a letter at the center of screen. The letter could have been C, M, P, S, U, corresponding to cinema, music, politics, sport, or unfamiliar. And the subject's task was to do a match-non-match task. Basically, they had to indicate whether the letter matches with the category of the face image or not. Again, one of the reasons that we use this design was to minimize motor confounds, because if we had associated each face with a certain response of the finger, whatever we would have found in the fMRI map may have reflected the motor representations. By using this design, which is a match non-match task, we basically dissociated representation of faces from motor representations. So we presented all 40 face images in each functional run. So we had the sequence of these trials. And then we repeated this process and we collected 16 functional runs from each subject. But the important thing is that in each run, we presented all face images, all 40 face images. We also had some blank periods between trials with random duration, so that we could basically separate the events more optimally with this design. We had 16 subjects. We collected data using Prisma scanner in NBML, and these are the sequence parameters. We had a resolution of 3.5 millimeter isotropic voxels. As I mentioned, we collected 16 functional runs from each subject, and we analyzed data using free surface software. So the first thing that we looked at was cortical representation of familiar faces. So here I'm going to show you a result of a univariate analysis. So we compared familiar versus unfamiliar faces. And this comparison showed regions in posterior cingulate cortex, anterior cingulate cortex, temporoparietal junction, and anterior lateral temporal cortex. So these four regions were activated by familiar faces, which is consistent with what people have shown in previous studies. So this is not a novel result. So it was basically a replication of previous findings. The activations were slightly stronger in left hemisphere. So the upper row corresponds to left hemisphere, the lower row corresponds to right hemisphere. And another important result here was that we didn't see any activations in ventral temporal cortex, even though people think that at least anterior and ventral parts of temporal cortex should be involved in familiar face processing, but we didn't get any activations in ventral temporal cortex, including some classic face areas such as occipital face area and fusiform face area. We also didn't have any activations in V1 in back of the cortex, which is good. It shows that we didn't have low-level differences between our face categories. So V1 could not distinguish familiar from unfamiliar faces. We confirmed that this pattern of activation is reproducible. So if we compare each familiar face category versus unfamiliar face category, we basically get the same set of areas. So these four sets of maps are quite similar to each other. Okay, so far I talked about one of the stages of familiar face processing. Now we are going to look at the cortical representation of familiar face subcategories. So it would be another abstraction level in the hierarchical processing of familiar faces. Here we are going to use a multivariate pattern analysis, namely searchlight analysis, to see which part of the brain contains information about subcategories of familiar faces. So in this analysis, which was basically a kind of surface-based searchlight analysis, we defined a small region on the cortical surface. I'm calling it a surface-based searchlight ROI. So it was a small region. We looked at the pattern of cortical activations in this region, and then we moved this region all over the cortex to basically scan the entire cortex. So this procedure is called searchlight analysis. And then how we look at the pattern information in each searchlight ROI, so that has been described here. Basically, what we do here is that we split the runs, the functional runs, into two groups. For example, odd runs and even runs. And then we look at the pattern of activations across voxels within this searchlight ROI. And then we compute the correlation of activation patterns between odd and even runs. And we come up with a correlation matrix. We do this procedure for all possible pairs. So what are our pairs? So we have cinema, music, politics, sport, and unfamiliar categories in the odd runs. And again, we have cinema, music, politics, sport, unfamiliar in the even runs. We compute the correlation of activation patterns. And then the logic here is that if there are reliable information about these subcategories, we expect that the correlation in the diagonal elements should be high, and the correlation in off-diagonal elements should be low. Basically, the patterns, so basically there are some specific patterns for each subcategory in a way that they are replicable across two sets of runs, and we can get a high correlation in the diagonal elements and low correlation in off-diagonal elements. So here I'm showing that procedure again. So for each searchlight ROI, we have a correlation matrix, and we compute the correlation of this correlation matrix with a model matrix. So we compute the correlation of this matrix with a model matrix. The model matrix is basically an identity matrix, so it has value of 1 along diagonal elements and value of 0 in off-diagonal elements. So we compute Kendall-Tau correlation, and if we get a high correlation, it means that that region contains information about familiar face subcategories. So we do this analysis for all possible searchlight ROIs, and we come up with a cortical map. So these maps show which parts of the brain contain information about familiar face subcategories. So what we found was that a region in posterior cingulate cortex within Brodmann area 23 had good information about familiar face subcategories. We also found 2 other spots in posterior parietal cortex and also in lateral prefrontal cortex. I should mention that Brodmann area 23 is one of the main subdivisions of posterior cingulate cortex. And also the activations were stronger in left hemisphere again. In another analysis which was related to the searchlight analysis, we looked at pattern information within some selected ROIs or regions of interest. We basically looked at some classic face areas, including occipital face area, fusiform face area, and anterior temporal face patch. This anterior temporal face patch is a region that is located further anterior in temporal lobe. And this is a region that I described it several years ago. It appears to be the homologue of one of monkey anterior face patches. And this region is thought to be involved in the processing of face identities. So we also included this region as one of the ROIs. And here I'm showing those correlation matrices for Brodmann area 23, anterior temporal face patch, FFA, OFA, and V1. And similar to what we found in the searchlight analysis, only Brodmann area 23 contained information about familiar face subcategories, and the other regions didn't have much information. We also use another approach to look at pattern information. Here in panel C, we use SVM analysis, support vector machine analysis. So in this analysis, we trained the classifier to distinguish different subcategories of faces based on the activation patterns that we had got. And then after training, we tested the classifier on the left out functional run. So basically, we trained the classifier with 15 functional runs and tested on the remaining functional run to see whether the classifier can distinguish and these 5 face categories based on the activation patterns. And again, Brodmann area 23 had a high decoding performance for all possible pairs of classification, for example, cinema versus music, cinema versus politics, and so on. And the other regions didn't have much decoding performance. We also looked at 2 subcortical ROIs, amygdala and hippocampus, because these 2 regions sometimes show activations for faces. So we also looked at these 2 regions, and again, they didn't have any information about familiar face subcategories. Here in the y-axis, we used a different metric to look at the pattern information. So instead of computing the correlation between correlation metrics and model metrics, another thing that we can do, we can calculate the difference between the values of diagonal elements and off-diagonal elements, and use it as a category discriminability index to evaluate pattern information in these regions. So the bottom line is that no matter how you define pattern information, only Brodmann area 23 contained information about familiar face subcategories. Okay, in the next stage, we are going to look at cortical representations for familiar face identities. So this would be another level of familiar face processing, which is a more specific level of processing. So the procedure was very similar to the previous one. We again did a searchlight analysis, and the only difference here was that instead of having a 4x4 correlation matrix, here we have a 32 by 32 correlation matrix. So we basically included all individual familiar faces in this matrix. So we are looking at the activation patterns for all individual faces. And the model matrix, the categorical, I shouldn't say categorical model, so basically the model that can distinguish identities of faces is a matrix like this. So it has a value of 1 along diagonal elements and value of 0 for off diagonal elements. And again, we compute the correlation of these two and visualize a cortical map. So what we found was that, again, a region within posterior cingulate cortex contained information about familiar face identities. And also we had regions in frontal cortex that contained information. So with this analysis, we basically looked at which parts of the brain could distinguish individual faces. We also looked at a more general level of face representation, which would be basically the representation of faces as compared to other object categories. So we want to see which parts of the brain just cares about faces, no matter whether they are familiar or unfamiliar. Basically, these regions separate faces from other object categories. So this is a very general level of face processing in the brain. In order to get a very high quality map, we used data from Human Connectome Project database, and we made a group average map of about 800 subjects. So this database is publicly available, and we just analyzed this data. And then we had a set of areas that were face selective. They showed more response to faces compared to other object categories, including places, tools, and body parts. And these are pretty much classic regions of face processing, including OFA, FFA, anterior temporal face patch, 3 regions along superior temporal sulcus. And interestingly, we found a relatively new area in posterior cingulate cortex again, which was face selective. Okay. So from all the analysis that we did, we found a region in posterior cingulate cortex which had some form of representation for faces. Either it was familiar face representation or familiar familiar face subcategory representation or familiar face or identities of familiar faces. So we had different kinds of representations in posterior cingulate cortex, especially in the left hemisphere. So here we are putting all of them together to look at the topographic relationship between different kinds of semantic face representations. So in panel A, we have the result from the last analysis, so comparing faces versus other categories. So we had this region in posterior cingulate cortex, and we have another region which came out from the comparison of familiar versus unfamiliar faces. And also we had regions that came out from searchlight analysis. And these are regions that contain information for either familiar face subcategories or familiar face identities. And then we compared the location of these regions in this map. So as you can see, all four regions had a lot of spatial overlap, but also at the same time, the peak activation seemed to be shifted. So even though these regions are overlapping, but they're also spatially distinct. This is just a control analysis to make sure that the smoothing that we used in the univariate analysis doesn't change the location of this activation. So in the univariate analysis, we had smoothing in the data. We repeated the experiment without smoothing, and we basically got the same activation. In the pattern analysis, we didn't use any smoothing at all because we wanted to preserve fine-grained information in the data. So as I mentioned, these four regions showed high spatial overlap as quantified by Dice coefficient. But importantly, the peak activation of these regions were significantly different from each other. So in this analysis, which is a little bit complicated, we basically quantified the spatial shift in the location of activations, and we found that the peak activation of these four regions are significantly different from each other. So if we quantified the distance between peak locations using a Euclidean distance, we find that the distance of peak activations are actually significantly different from each other. So I'm going back to this slide again. So the important finding here is that there seems to be some adjacent regions in posterior cingulate cortex which process distinct properties of familiar faces. And these properties are relatively hierarchical. So some of them are more general information and some of them are more specific information. Finally, we looked at the connectivity patterns of these four regions, these four PCC regions. So in this analysis, we defined seed regions within posterior cingulate cortex. So we had four seed regions. And then for each of them, we computed the connectivity map based on resting state data. So for each seed region, we had a whole brain map of regions which were connected to this seed region, okay? We did this for all four seed regions. And then we used a winner-take-all procedure to combine these connectivity maps. So after the winner-take-all procedure, we can see which parts of the brain are preferentially connected to each of these seed regions, okay? So we get a map like this. And as you can see, it looks like these four regions have somewhat distinct patterns of connectivity. Another result here is that anterior temporal face patch in basically, yeah, it should be here, in ventral temporal cortex is preferentially connected to one of PCC regions which responds more to familiar faces. Basically, this is the region that distinguishes familiar from unfamiliar faces. So I can skip these points. So basically, what we found was that there are some spatially adjacent regions within left posterior cingulate cortex that process distinct properties of familiar faces. And we didn't get any activations in anterior temporal lobe, even though in the literature ATL has been mentioned as one of the main regions for familiar face processing. We didn't get anything in anterior temporal lobe, partly because we had some like data acquisition problems. So usually anterior temporal lobe is one of difficult regions in Fmri. We usually have low signal to noise ratio in that region. So in order to get a good signal from ATL, you need to use some spatial MRI sequences. So hopefully with a more optimized imaging technique, we can get a better signal from ATL, and then we can evaluate whether ATL contains information about, let's say, familiar face subcategories or not. At the end, I want to mention one of the implications of our findings, which is in Alzheimer's disease. So if we look at the pattern of pathology in Alzheimer's disease, so here I'm showing you 3 maps. The first one is the map of cortical atrophy. The second one is the map of hypometabolism, and the third one is the map of beta amyloid deposition. So as you probably know, in Alzheimer's disease, medial temporal cortex and hippocampal region show cortical atrophy, and this is where the pathology starts. And then in the advanced stages of Alzheimer's disease, we have the involvement of other brain regions, including posterior cingulate cortex, which is quite interesting. I mean, this map is quite similar to the map of familiar versus unfamiliar faces, which suggests that perhaps one of the reasons that AD patients cannot recognize their relatives, their close relatives or friends, could be because of the involvement of PCC. So basically, the PCC region has been affected and they cannot process familiar faces as they should. Okay, thanks for listening, and this is the picture of IPM, a very beautiful place. Of course, Neda did her PhD in another building, not in this building, but anyway, this is a nice, a very nice place to work, actually. Okay, thank you. So if you have questions, we can talk about those.
Thank you, Dr. Rajimehr. It was very interesting talk and topic. We have, I think, enough time to have few questions. Can I ask you if you have any idea about, I mean, at the beginning you mentioned the different areas that are involved in face processing. I'm wondering whether these areas are different for different topic of processing, like detection, recognition, or the one you mentioned, like emotion processing of faces, for example, or something like that. Or we expect that these areas are different, they should be different, or maybe they are the same when we are processing faces, but maybe the way that these areas are communicated are different. I mean, the areas are the same, or they can be different for different type of processing.
So regarding these face areas, I think we have good knowledge about their function based on what people have done in monkeys. So in fMRI, we can't really address the coding questions. We can only look at where in the brain we have some sort of representations. But if we look at the homologues of these regions in monkeys, and there have been a lot of nice studies by Doris Chow and Werner Freibad. They have looked at these face areas, and actually the response properties are sometimes different. So it's not just a matter of connectivity, and their selectivity, their response profile is different. For example, the middle face patch in monkeys, which corresponds to FFA, it looks like it's more involved in face detection, but as we go more anteriorly in anterior face patches, we have, like for example, in face patch AM, we have view invariant identity representation. So it looks like that region cares more about face identification. Also regions more dorsally in superior temporal sulcus looks like they encode facial expressions. They are highly sensitive to dynamic faces and also to expressions of faces. So it looks like their response properties are actually different. Yeah.
Thank you. Okay, Dr. Rajizadeh and Dr. Sanagi, both have question. Dr. Rajizadeh, you can go ahead.
So, hi Reza, thanks for the good talk. The result that you showed was interesting for me, especially the analysis of the Human Connectome project, because I think for the first time I could see face patches in human brain that kind of, as you mentioned, paralleled the face patches that we know in monkey.
Exactly.
And for me was very educational. But one thing that I wanted you to comment on is that the PCC, as far as I know, as you said, like you would have expected to see these more advanced analyses in more anterior patches like ATL. But you kind of discovered or you found these things in PCC, which is, as far as I know, in monkey, a cingulate cortex, or maybe you can correct me if I'm wrong, a cingulate cortex is not really involved in face processing. So what is your take on this region? Is this, does this, first of all, have any homologue in monkey? And if it does, where it is? If it doesn't, why this area all of a sudden appears in humans?
So yeah, very, very interesting question. I actually communicated with Winrich Freibad about this PCC region, and I had some conversations with him. So first of all, there seems to be some debates whether there is even PCC region in monkeys or not, or whether we have the homologue of human PCC in monkeys. And I specifically asked him whether this PCC region shows up in the fMRI of monkeys, because he has done an fMRI study on familiar face processing in monkeys, and he had a paper in Science, and they didn't show a medial view of cortex, so I wasn't sure whether there was an activation there, but was just not reported. And he actually confirmed that he didn't find any activations in PCC of monkeys. If we think there is PCC, he didn't find any activations in this kind of location in cortex of monkeys. So what's going on here? There might be species-specific differences. So PCC region in humans is more generally involved in semantic processing and also extracting information from memory and also in representing contextual associations. It's possible that humans have more contextual associations, they have richer semantic representations, and that's why we get some activations in PCC, but in monkeys we don't get that. The other possibility is that in order to get an activation in the PCC of monkeys, we need to use a much richer stimulus, like for example a movie stimulus that engages monkeys in some sort of semantic processing. If you just show some like static images of monkeys that are familiar to the subject monkey, and that may not be sufficient to drive activations in PCC. But in any case, like this region of cortex is one of difficult regions to record from, so we don't really have much data from PCC of monkeys. We know that this region is part of the default mode network in monkeys, similar to what we have in humans. So there are some similarities in the organization of PCC in humans and monkeys. But what this region is exactly doing in monkeys, we don't know much about it. Yeah.
Thank you very much.
Okay, thank you. Please.
Hi Reza, it's nice to see you again.
Hey Mehdi, how are you?
I'm fine, thank you. I have, I think, two questions. Sorry if I'm not very clear, but to me is that the PCC is part of the limbic system, am I right?
Yes.
And my second question is, I mean, all these familiar faces, they're also from famous people.
Let me just mention, just let me mention an interesting thing. So I was reading a paper from Pessoa. He's a main figure in, I think, affective neuroscience and emotion processing. The term limbic seems to be one of useless terms in neuroscience because it refers to many heterogeneous functions, and actually he argues that it's quite unhelpful when we use this limbic term. But yes, classically PCC is part of limbic lobe, at least, if we don't say limbic system. Okay.
I'm sorry for my lack of knowledge about the human anatomy. But my second question is kind of prime. My second question is that all these faces that you show subjects are from famous people, which are also celebrities. And celebrities is a kind of, I would say, human concept. And they're not only familiar, but they also have some other meanings to them. Maybe this is what you meant by semantic. And what's your thought about this kind of way of thinking?
Yeah, that's a good point. People have often distinguished familiar face processing from famous face processing. So, sometimes a face is personally familiar to us, like the face of a mother or sister. And sometimes the face is not personally familiar, but it's just famous, okay? I mean, in both cases, we have a lot of semantic information about these faces, yes. But the nature of semantic information might be different. And actually, some people have shown that when you do Fmri and when you compare representations for familiar versus famous faces, there seems to be some distinctions. So like some regions in prefrontal cortex care more about familiar face processing, and some regions care more about famous face processing, and also that's true in the PCC region. But both of them also share a lot of processing, because in both cases we have some sort of semantic representations. Did I answer your question, or?
Yes, yes, thank you. Thank you very much.
Okay.
Okay, thank you. Any other questions? Can I ask another question in line with the question of Dr. Rajzadeh Ali? As I know, I mean, this area you mentioned that is a new MFA, is a new area that you have seen from the analysis you have done in human connectome data. Do you know
It's
what?
relatively new. I mean, it has been mentioned in the literature.
Ah, okay.
It hasn't been studied much, I would say.
Okay, I wanted to ask if there is any difference between the analysis you have done with different, I mean, with the other studies that they haven't found this area. I mean,
yeah, that's a good point. Like in this map, it shows up because of two reasons. First, we have a lot of signal averaging, and the second is that in human connectome there is a very good cross-subject registration, so the alignment of subjects is very optimized because they use a multimodal registration. So sometimes in some group average maps you don't see an activation because of a bad registration, but in HCP they have a good alignment, and that has helped to identify this region. So with a lot of averaging and with good alignment, you get this activation. So perhaps one of the reasons that you don't see it in some of Fmri studies is because of like low number of subjects, and I mean phase selectivity here might be weaker compared to like FFA. So you need more averaging, you need a better alignment to get this activation. And also some people just assume that everything should be in temporal cortex. They don't even have a slice to cover this region, or even if they have slices, they don't even look at this region. They just think this region is just junk. Let's go and look at temporal cortex. But yeah, we also have this activation here, which has actually been reported long time ago by Jim Haxby and colleagues like Ida Gobini. They actually reported this region as part of extended face processing network, but it hasn't been studied much, I would say.
okay.
There are some studies on familiar face processing in this region, but generally this region is somewhat ignored, I would say.
Okay, thank you. Okay, so as